Structure and function of the musculoskeletal system. Bones do more than provide structure. They are essential for multiple body functions. They support tissues, protect vital organs, and permit movement by serving as attachment points for muscles. Bones also play a critical role in blood cell formation and in maintaining mineral and hormonal homeostasis, especially for calcium and phosphate. Without healthy bones, the entire musculoskeletal and metabolic system would be compromised. Bone tissue is a specialized form of rigid connective tissue. It consists of bone cells, collagen fibers, ground substance, and crystallized minerals. The minerals give bone its strength, while the fibers and ground substance provide a supportive framework. This composite structure allows bones to be strong, flexible, and resilient. Bone cells are responsible for the dynamic nature of bone. They help it grow, remodel, and repair. The bone matrix provides the scaffold where mineralization or calcification occurs. This process strengthens the bone and helps maintain skeletal integrity. Without it, bones would be weak and brittle. There are three main types of bone cells: osteoblasts, osteocytes, and osteoclasts. Osteoblasts are bone-forming cells that synthesize osteoid, the unmineralized matrix. They also produce osteocalcin and express RANKL, which helps form osteoclasts. In contrast, osteoclasts are large, multinucleated cells that break down old bone tissue during resorption. Osteocytes are mature bone cells that originate from osteoblasts. They become embedded in the bone matrix and play a central role in both bone formation and bone resorption. These cells are crucial for sensing mechanical stress and signaling bone remodeling. Their activity helps maintain the strength and health of the bone throughout life. The OPG/RANKL/RANK system is key in regulating bone turnover. RANKL, produced by osteoblasts, binds to rank on osteoclast precursors, promoting their maturation and increasing bone resorption. Osteoprotegerin acts as a decoy receptor for RANKL, preventing excessive bone breakdown. This system helps balance bone formation and bone loss. The bone matrix is primarily made of collagen fibers, which provide tensile strength. Proteoglycans are also present, helping to retain calcium and support calcification. These components interact to give bone its flexible yet sturdy nature. The quality of the matrix is vital for preventing fractures. Glycoproteins help organize the collagen fibers and regulate mineralization. Key proteins like sialoprotein, osteocalcin, and bone albumin all contribute to bone structure. Bone mineralization occurs in two phases: initial crystal seeding, followed by crystal growth. Calcium and phosphate are the primary minerals involved. There are two main types of bone tissue: compact or cortical, and spongy or cancellous. Compact bone makes up 85% of the skeleton and contains the Haversian system, a highly organized structure of canals and layers. Spongy bone lacks these systems, but consists of trabeculae, which provide strength with reduced weight. Both types are essential for skeletal function. This slide visually compares compact and spongy bone. Compact bone is dense and forms the outer shell of bones, while spongy bone is porous and found at the ends of long bones and in the interior of flat bones. The periosteum, a fibrous membrane, covers bone surfaces and contains nerves and blood vessels. This membrane supports bone growth and healing. The human skeleton is made up of 206 bones, divided into two major sections: the axial skeleton and the appendicular skeleton. The axial skeleton consists of 80 bones, including the skull, vertebral column, and thorax. The appendicular skeleton contains 126 bones that make up the limbs and the girdles that attach them to the axial skeleton. Together, These structures provide support, protection, and mobility. This anatomical image highlights the distinction between axial and appendicular bones. The axial skeleton provides the body's central framework, while the appendicular skeleton enables movement. Both divisions work together to maintain posture, protect organs, and allow for locomotion. Proper alignment and balance of both systems are essential for functional biomechanics. Bones are classified by shape: long, short, flat, and irregular. Long bones, like the femur and humerus, contain a diaphysis, metaphysis, and epiphysis, with growth occurring at the epiphyseal plate. These bones also contain a medullary cavity, which houses bone marrow. The endosteum lines the internal bone surfaces and plays a role in bone repair. This illustration shows internal bone anatomy, including the compact outer layer and the spongy inner layer. Long bones are designed for leverage and weight bearing. The medullary cavity contains yellow or red marrow, depending on age and function. Blood vessels run through the bone to nourish cells and support remodeling. Flat bones, like the ribs and scapulae, consist of two plates of compact bone with spongy bone between. Short bones, such as those in the wrists and ankles, are cube-shaped and provide support with limited motion. Irregular bones, including the vertebrae and facial bones, have complex shapes suited for specialized functions. Each bone type plays a unique role in overall skeletal mechanics. Bone health depends on continuous remodeling, a process that replaces old bone with new. Basic multicellular units coordinate this cycle. Remodeling has three phases: activation, where cells prepare the area; resorption, where osteoclasts break down bone; and formation, where osteoblasts lay down new matrix. This process maintains bone strength and responds to stress and injury. This diagram illustrates the dynamic process of remodeling. Microscopic injuries are repaired through careful coordination between bone resorbing and bone forming cells. The balance of this activity is critical. Too much resorption can lead to osteoporosis, while inadequate resorption can result in dense but brittle bones. Hormones like PTH, vitamin D, and calcitonin regulate this cycle. Bone injuries heal in stages similar to soft tissue, but the final product is new bone, not scar tissue. The process includes hematoma formation, procallus formation, callus formation, replacement, and remodeling. Each step is essential to restore both structure and function. Adequate nutrition, vascular supply, and mechanical stability support successful healing. A joint, or articulation, is the site where two bones meet. Joints are classified by both structure and function. By movement, joints are either synarthrosis or immovable, amphiarthrosis or slightly movable, or diarthrosis or freely movable. The type of joint determines the range of motion and stability provided. Joints can also be classified by their structural composition. Fibrous joints connect bones via dense connective tissue and allow little to no movement. Cartilaginous joints are connected by cartilage and offer slight mobility, such as the pubic symphysis. Synovial joints are the most movable, featuring a joint capsule, synovial membrane, and articular cartilage. This anatomical illustration shows the different types of structural joints. Sutures and syndesmoses are examples of fibrous joints. Cartilaginous joints include symphyses and synchondroses, while synovial joints come in multiple configurations: uniaxial, biaxial, or multiaxial, allowing a wide range of movement. Each type provides unique support depending on its anatomical location. The joint capsule is a fibrous covering that encloses the joint. Inside, the synovial membrane lines the capsule and produces synovial fluid, which lubricates and nourishes the joint. The joint cavity allows movement between bones, while articular cartilage covers the bone ends, absorbing shock and reducing friction during movement. This close-up image shows the key components of a synovial joint. Synovial fluid is a clear, viscous substance that resembles egg white and helps cushion joints. Articular cartilage is composed of hyaline cartilage and lacks blood vessels, relying on diffusion for nutrient delivery. Its smooth surface enables frictionless movement. Synovial joints are highly specialized for mobility and weight distribution. Their features allow for smooth, controlled motion in the body's limbs and spine. Disorders affecting these structures, such as arthritis, can significantly impair function. Understanding these components is key to diagnosing and treating joint-related conditions. Skeletal muscles are composed of millions of individual fibers that contract and relax to produce movement. They make up about 40% of adult body weight and are responsible for maintaining posture, generating heat, and stabilizing joints. Muscle tissue is rich in water, protein, and metabolic compounds. Muscles respond to both voluntary control and reflexive stimuli. There are more than 350 named muscles in the human body. Fusiform muscles are long and strap-like, optimized for range of motion. Pinnate muscles are broad and fan-shaped, suited for generating power. Muscle structure varies depending on its function and location. This slide outlines muscle organization from outermost to innermost. The epimysium surrounds the entire muscle and merges into the tendon. Perimysium groups fibers into bundles called fascicles, while the endomysium wraps each individual muscle fiber. This layer design allows for strength and flexibility during contraction. Skeletal muscle is voluntary and striated, controlled by motor units composed of motor neurons and the muscle fibers they stimulate. Muscle spindles and Golgi tendon organs are sensory receptors that help regulate stretch and force. Each unit responds in an all-or-nothing fashion, meaning it either contracts fully or not at all. The pattern of recruitment determines the strength of movement. Skeletal muscle fibers are voluntary, striated, and known as extrafusal fibers. The motor unit is the basic functional unit of the neuromuscular system, consisting of a motor neuron and the muscle fibers it controls. Fine motor control, like in the fingers, involves many small motor units. Larger muscles, like the quadriceps, require fewer but larger motor units for powerful contractions. This diagram shows how motor units connect the nervous system to skeletal muscle. A motor unit consists of a motor neuron from the spinal cord and all the muscle fibers it innervates. Each axon branches into multiple neuromuscular junctions, allowing one neuron to activate multiple muscle fibers simultaneously. Small motor units control fine movements like those in the fingers, while large motor units control powerful muscles like the quadriceps. Each muscle fiber is a single elongated cell containing myofibrils, which are the contractile elements. Muscle fibers originate from myoblasts, the precursor cells responsible for muscle growth and regeneration. Satellite cells are dormant myoblasts that activate during muscle repair. These cells play a key role in healing after injury or strain. Muscle fibers are categorized as white type 2 and red type I fibers. Type 2 fibers are fast twitch and used for short bursts of power, while type I fibers are slow twitch and fatigue resistant, ideal for endurance. Each fiber contains a sarcolemma and basement membrane, and the sarcoplasm stores enzymes, proteins, and oxygen-binding molecules. These components support contraction and energy production. The sarcotubular system includes the transverse T-tubules and the sarcoplasmic reticulum. These structures regulate calcium storage and release during contraction. Calcium is essential for initiating the interaction between actin and myosin filaments. Efficient signaling within this system ensures coordinated muscle contraction. Myofibrils are the contractile units of muscle fibers and are made up of repeating units called sarcomeres. The speed and strength of muscle contraction depend on how quickly sarcomeres shorten and lengthen. Myofibrils are the most abundant subcellular structure in muscle cells. Their integrity is crucial for overall muscle function. This slide visually summarizes the architecture of skeletal muscle fibers. Sarcomeres contain myofilaments, primarily actin and myosin, which slide past each other during contraction. The precise arrangement of these filaments gives skeletal muscle its striated appearance. Proper alignment and function of myofilaments are essential for muscle strength and flexibility. Muscle metabolism also involves non-protein molecules such as creatine, phosphocreatine, purines, and uric acid. These substances play roles in energy storage, buffering, and waste elimination. Glycogen is a major energy reserve within the muscle. Electrolytes and minerals help regulate protein synthesis, enzyme function, and cellular stability. Muscle contraction begins with excitation, when a nerve impulse triggers a muscle fiber action potential. This leads to coupling, where calcium is released, followed by contraction, where actin and myosin interact to generate force. Relaxation occurs when calcium is reabsorbed and the muscle returns to its resting length. The process is known as the cross-bridge cycle and is powered by ATP. Muscle activity requires a constant supply of ATP, which increases up to 100-fold during exercise. Short bursts of activity rely on anaerobic glycolysis, while sustained contractions depend on aerobic metabolism. Type I fibers are more fatigue-resistant due to their oxidative capacity. Maintaining glucose and oxygen delivery is key to performance and recovery. Skeletal muscles follow the all-or-nothing principle, where each motor unit contracts fully when stimulated. Repetitive discharge of motor units creates smooth, controlled movements. When stimulation is rapid and the muscle cannot relax between contractions, it reaches physiologic tetanus, producing maximal force. The number of motor unit recruited affects the strength of the contraction. Muscle contractions are categorized as isometric or dynamic. In isometric contractions, the muscle maintains constant length while tension increases, like holding a plank. In dynamic contractions, the muscle changes length while maintaining tension. Dynamic contractions can be concentric, where the muscle shortens, or eccentric, where it lengthens while resisting force. This visual highlights the mechanical behavior of muscles during movement. Muscles act as agonists to create motion and antagonists to control or resist it. For example, when you bend your elbow, the biceps act as the agonist and the triceps as the antagonist. This balance allows for smooth, coordinated movement. Tendons attach muscle to bone and serve as biological springs, transferring force during movement. They also help stabilize joints by resisting excess motion. Ligaments connect bone to bone and contribute to joint stability. Both structures are made of dense connective tissue and play vital roles in mobility and injury prevention. As we age, bone mass declines, making bones less dense, more brittle, and slower to repair. Joint cartilage becomes stiffer and more fragile, reducing flexibility and increasing the risk of joint degeneration. These changes result in a reduced range of motion and a higher risk of fractures. Maintaining bone health is critical in aging populations. Sarcopenia, the loss of muscle mass and strength, is a hallmark of aging. This leads to decreased oxygen use, a lower basal metabolic rate, and reduced physical function. Combined with bone loss, sarcopenia increases the risk of falls and frailty. Resistance training and proper nutrition can help slow this decline.